Welcome back to our next episode of Chromatography Experts, a podcast that talks about all things separation science. I'm your host, Abbie Breton, Manager of Application Development here at Tosoh Bioscience, and I'm thrilled to be joined today by my colleague, Yuliya.
Hi, I'm Yuliya McAnany. I'm an application scientist for process resins with Tosoh Bioscience.
So today we'll be diving into all things adeno-associated viruses and also some cell and gene therapy. And we're going to be spotlighting a recent method, a recent breakthrough method that we found here at Tosoh. So let's dive right in. Hey, Yulia, welcome. Excited to have you. For listeners who may be new to gene therapy, can you just give a quick explanation on what AAVs are and why they're such popular vectors?
Gene therapy is a new medicinal modality to treat genetic disease mostly. So, AAV stands for adeno-associated virus. This is basically a technology for delivering DNA, so nucleic acids, into a cell. So what happens is you would have for example, a patient with a defective, let's say enzyme that's causing disease. So what you can do is you can replace or correct the gene that is defective, and hopefully ameliorate the disease. The reason that AAVs are popular and why they've become kind of the workhorse, very popular vector is that AAV is not a naturally pathogenic virus, so it does not have a pathogenic function in humans. So what that means is that people generally tolerate the administration of this virus at appropriate doses. The other thing is that um, it is well suited for in vivo administration because of its non-pathogenic features. So what you can do is you can take this AAV, you can replace almost all of the genome, the natural genome inside it, except for the necessary structural elements. You can replace it with your gene of interest. So this can be, for example, the transgene for the effective enzyme that the person might be missing or is non-functional. You can replace it with an engineered version that has various features. So hopefully that would fix the disease. The other thing is that AAV is a very diverse class of viruses. So there are multiple serotypes. And the advantage of having multiple serotypes is that uh, it has tissue targeting potential. So there are serotypes that preferentially transduce brain tissue. There are serotypes that preferentially transduce muscle or liver tissue. You can have also engineered capsids that are not natural, but they have the features that you wanted, such as stability, um, uh, and tissue targeting.
Okay. So you said that, at appropriate doses, they are non-pathogenic. So is there anything so there's nothing that would make these pathogenic to in a human. There's no sorts of like impurities or anything that we're looking at when we're looking at AAV. So if you think about like how MABs, there's some things that like we're looking to when we purify MABs that we're looking to like purify out of those. So when we look at AAVs, is there something we're looking at here to purify out when we produce them?
Right. So because AAVs are biologics, they're still going to have these product related impurities and process related impurities because they are produced inside host cells, first of all. So there you still would require the purification of host cell DNA and host cell protein. So yes, like a high degree of purity is required, and that's why it's necessary to use chromatography for them. The other thing is that although they're not pathogenic, they're still immunogenic at high doses. So the capsid protein is still a foreign protein. Any escape DNA that is also foreign DNA, and at very high doses, the either you can have a patient with pre-existing anti-AAV antibodies that would prevent them from being dosed with this because they will just mount an immune response immediately and eliminate the capsids. But also a person might develop an immune reaction of serious immune reaction that might cause bad side effects such as liver damage. So one of the product related impurities for AAVs would be empty capsids.
Yeah, and so why are those so those are so critical, like, why are those so critical, those empty versus, like, let's say, like, a full capsid?
Right. So the full capsid actually contains the gene of interest. The full capsid contains the replaced genome that has this engineered transgene in it. So that's actually the therapeutic material is the gene. An empty capsid devoid of DNA has no therapeutic material. So if you dose someone with a formulation with excessively high empty capsids, you're basically stimulating the immune response for no reason. So basically, you want as many shots on goal as you can have by having as many of the capsids be as full as possible. So that would basically hopefully increase the potency of your drug and minimize the immunogenic potential.
Understood, yeah, so I imagine, like, when you produce these, you are going to get a mix of empty and full, and you're just trying to get rid of as many of those empty capsids as possible.
Right. So the process of AAV genome packaging is just inefficient naturally, especially with this triple transfection method. So often you would see filling ratios across the board from like 10 to 50% full. So quite a broad spread.
Yeah.
Hmm.
Okay. And, like, so what are some of the challenges that you face and scientists face in, like, when you go to purify that, removing those empty capsids? I imagine there are quite a few challenges there.
Sure. So, number one is selecting a scalable method. So, a method that works at the research scale is not necessarily going to translate into being a production commercial scale. So, for example, preparative ultracentrifugation. For example, preparative ultracentrifugation that is a method that cannot be scaled up. You can only process a maximum of 2-ish liters with this method. So if you have a multi like hundreds of liters batch, you cannot possibly purify this. So this is an example of a method that cannot be scaled up. It can only be scaled out. You can only purchase more ultracentrifuges and obviously that's not appropriate for a clinical scale. So selecting a scalable method first when you're in development is something that I would recommend, but it's a big challenge when you're trying to you identify a molecule that would be effective, but you don't have the purification scheme set up for it.
Mhm. Yeah, for sure. So speaking of a scalable method, I hear that there's a recent publication that came out from Tosoh. Um, I would like for you to give us some insights into that publication. You give us the title and just an overview of what was in that paper.
Sure. So this publication in Journal of Chromatography A, it's called Preparative Choline-based anion-exchange chromatography for enrichment of full adeno-associated virus capsids. So, um, basically, to give you some background, so previously we had a paper describing the use of analytical anion exchange chromatography using choline chloride as the salt. So choline chloride is a non-toxic salt because of its favorable safety profile and also the fact that it's also available in GMP grade already because it's used as a supplement in the various supplements. So because it has a favorable safety profile, we thought that maybe this would be a good choice for a preparative anion exchange chromatography for AAVs to separate empty from full, so a polishing method. So what we did was we compared, for example, the performance of choline chloride versus traditional metal salts such as sodium chloride. We saw over a twofold increase in resolution when you use choline chloride compared to an equimolar concentration of sodium chloride. So we basically went through a full purification optimization scheme and I gave examples on how to optimize from a linear gradient to a step gradient that's customized for a particular serotype So the serotypes we tested were AAV5 and AAV8, and so I designed a method to purify polished AAVs and enrich fulls.
Nice. Okay. And what were some of your results? So I imagine, when we look at mab purification, we're looking at recovery, we're looking at like purity of that. So we're talking about with this, we're looking at separating out all those empty capsids and, enriching those full capsids. What kind of results were we looking at here for like full capsids and recovery numbers? And sorry, and also like what is typical, I would say, for AAVs? I know those are obviously different molecules than MABs. So if you can just give us those.
Right. So basically, people are aiming for at least 70% fulls in their final formulation. So that is something that a metric that regulators have suggested to have fewer than 30% empty capsids. And the benchmark for recovery, I would say, is about 50%. If you can get 50% for your yield, that's acceptable. Um, what I was able to achieve, we were able to achieve about 90% full capsids with 75% recovery. So, compared to these traditional polishing methods, I would say it's far better.
I mean, yeah, it sounds like those are like that's really far better than what we were like what industry is currently at, I would say.
Yeah, I would
Yeah.
that's been my experience is that this achieves it goes beyond the target
Yeah.
for sure.
Yeah, for sure. And do you know, I mean, it's just kind of offhand, do you know like what your starting sample was? Like if you're looking at like what your starting full concentration was? I'm just interested to see like how much enrichment you had there.
Right. So, talking about enrichment, people often talk about this enrichment factor. I don't think that's the best way to quantify the performance because it's so dependent on the start concentration. So, for example, most of the experiments had a about 50% full ratio to start with after affinity purification. So that's how I was able to get the 90% full capsids. However, there was an example with AAV5 where we had a very poor quality sample. So this would be like a less than 10% full sample. We were able to change that from 10% full to 83% full in one step. So, such a high enrichment factor is also really advantageous when you're talking about poor upstream feedstock content. So, you might have a really challenging sample where the expression is low, the filling ratio is low. This can also especially happen with very large genomes. If you're approaching the maximum packaging capacity, you might get lower filling ratios, and there might be nothing you can do about it on the upstream side. So, also, if you have significant batch to batch variability in your upstream feedstock, um, what this method can do is we demonstrated that it works on a broad range of different filling ratios, like upstream filling ratios. So, you you're not as hampered by a bad upstream batch with a poor filling quality. So what this means is that you could potentially reduce the total number of polishing steps that is necessary because this method is so effective, and that would translate into direct cost savings because it's so robust.
Oh, I mean, that's amazing. So during your research, like when you were optimizing this method, was there any like kind of like light bulb moments where you realized like, oh my gosh, we kind of made a breakthrough here with this purification?
I wouldn't say there was a particular moment, but over the course of these optimizations that I spent a long time doing, getting greater resolution with minimal empty capsid contamination in the full peak, that was pretty gratifying. Seeing the peak get sharper and taller every time, that was
Yeah.
pretty cool.
No, that's awesome. That's so exciting. So let's just kind of talk about like the broader implications of this new method for cell and gene therapy. So in the beginning, we talked about one of the biggest things is finding a method that's scalable. And so, how scalable is this method? I imagine you probably started this on like 1 mil columns and like scaled up a little bit. Is this scalable to like a clinical and commercial manufacturing?
So absolutely, very much so. So This study was done using the GigaCap Q650S. It was done using Toyo Pearl GigaCap Q650S in a skill pack column format. So these are pre-pack columns. They come in several capacities. For my work, I use the 1 mil and 5 mil skill pack. However, there are several other sizes. So they also come in 50 mil and 200 mil sizes, as well as bulk resin available for those who want to pack their own columns and custom column packing. So, the capacity for this resin is also very high. So, I was able to get 2.6 times 10 to the 14th viral capsids per mil of this resin. So it's rather high capacity as well. So you'd be able to purify a lot of it at once.
Yeah, yeah. So you could scale this up to like a clinical or commercial scale
Absolutely,
for sure. Yeah.
yes.
Okay.
Mhm.
No, that's really great to hear. And then kind of with that, could this be adapted to other viral vectors? I know we this paper focuses on AAVs. It's really the focus of this conversation today. But is this something that we could look at for other viral vectors?
Absolutely. So I think other viral vectors, other gene therapy vectors such as lentivirus, adenovirus, anything that has, you can also broaden this to non-viral delivery vectors, potentially with anything that has an encapsulated nucleic acid, as long as the outside of the capsid or whatever delivery vehicle you have has enough charge on it. It is pretty basic. Like that would maybe not basic. I don't know. Maybe maybe not. As long as long as, there's enough, like, encapsulated, DNA within, it's very possible that you could separate that, using this method.
Okay, no, that's interesting. And so let's just kind of talk about like the future of cell and gene therapy, just like AAV in general. So you've obviously spent a lot of time working on this paper. I know here at Tosoh you do a lot of work with AAVs. Is there any advice that you would give to researchers that are trying to optimize their workflow, specifically AAV workflows?
So I would start development with scalability in mind. Even if you're an academic research group or a startup, you don't have to just stick with, for example, preparative ultracentrifugation. It's very simple to get these chromatographic methods up and running, even without like the most sophisticated equipment. So putting in chromatography into your workflow early on with scalability in mind is something I would definitely recommend.
Yeah, no, and I think like most of the scientists that are probably working in this are so used to doing chromatography for whether it's for like MABs or other biologics that it's like an easier path than, like the ultracentrifugation. So I can see that for sure. Like, looking at that.
Yeah, there are major benefits, and one of the main ones is reproducibility. So with, for example, preparative ultracentrifugation, there's going to be operator variability because it has this manual intensive band collection step that's not really standardizable. It would be one individual pulling this band through a vial. That's not a really robust kind of procedure.
Yeah.
So with chromatography, you don't really need to worry about that. Your method will run pretty much the same way every
Yeah.
time. Yeah.
Yeah. So, we're at the end of 2025 here in this episode. So obviously 2026 is coming up. Is there anything new that you're working on in this AAV space? Anything you want to kind of like tease here?
Sure. So I'm interested in exploring even more improved gradients. So, um, just tweaking it even more. We developed this as kind of a case study. So basically seeing how far we can take this method, how we can change it to, for example, to deal with some of the more challenging serotypes, ones that are traditionally more difficult. For example, we didn't do this for AAV9, but I would be interested to see how AAV9 can perform here, because that's a notoriously challenging serotype to purify. The other thing is that, we've shown that this is pretty effective, but it would be even more impactful if we had an idea of the mechanistic understanding. So why is choline causing this improved resolution beyond just the chloride that is in there? So yeah, mechanistic understanding.
So thank you again for your time on this episode. That was really great information. It sounds like a really awesome method. And thank you all for joining us in this episode of Chromatography Experts. If you're working with AAVs or exploring other types of gene therapy, this episode's hopefully packed with a ton of insights for you as well. Be sure to check out the paper. It's open access. It'll be linked in our podcast notes, and we'll see you on future episodes.